Hello and welcome to our video lecture on section 15.5 on ribosomes and protein synthesis. So we've got a lot to talk about here, and our learning objectives are to describe the different steps in protein synthesis and discuss the role of ribosomes in protein synthesis. So protein synthesis is the same thing as translation, and it's our process of decoding the mRNA strand into a polypeptide product. And to do this, we're going to utilize those amino acids, and we are going to covalently string them together by interlinking peptide bonds. And this could be anywhere from 50 to 1,000 or more amino acid residues all strung together. So the way that we form these peptide bonds is if we were to take these two different amino acids, the R's, of course, would be their variable region that makes them the amino acid that they are. But we're going to focus on them having this carboxyl group and the amino group come together. So they are going to essentially become catalyzed by a ribosome, and by pulling these molecules, the two H's and the O, out, we are going to form a peptide bond between the carbon and the nitrogen here, and we get a water molecule as a result. So what we're going to need is our protein synthesis machinery. That's going to include our mRNA template that we created through transcription and was processed, our ribosomal RNA that it makes up 50% of our ribosomes. We need our transfer RNA and some various enzymatic factors. So we'll start off describing our ribosomes. This is going to be a complex macromolecule. It's composed of structural and catalytic rRNAs and other polypeptides. And we find these within the cytoplasm and our rough endoplasmic reticulum. We also have special ribosomes within our mitochondria and our chloroplasts, which are going to be of a different weight. So the ones that we're going to utilize in the cytoplasm for translation will be made up of a large subunit, which is 60S, and then the small subunit is going to be 40S, which when they're combined, it is 80S. And no, I'm not doing my math wrong. Note that these are not, I guess, accumulative, where if I added them together, it would be 100S, but this is used in a different type of units known as Svedberg units. So they are not additive. And so why do we need these subunits? The large subunit is going to help to bind the tRNAs. So you can kind of see that displayed over here. It has these little slots within it where the tRNAs are going to go and insert within it. And then our small subunit is going to really help to bind that mRNA template so that we can read it as we bring in the tRNAs and attach amino acids to them. And our ribosome is always going to read the mRNA strand from the 5' end to the 3' end and create the polypeptide from the N terminus, so where we saw that amino group, to the C terminus, where we saw that carboxyl group. And essentially these two subunits are going to stay together when they're synthesizing proteins and will dissociate when they are not synthesizing proteins. But when we have them together, we are going to call them a polyribosome or polysome. Now let's talk about our transfer RNAs. These tRNAs are going to be bound to a specific codon on the mRNA strand. And like we said, they have this corresponding anticodon that then is going to code for a specific amino acid that attaches to create our polypeptide chain. And so these tRNA molecules are helping to translate the language of our RNA strand into our language of proteins. And you could see that pretty well down over here. And we're going to have 3 factors that it interacts with in order to get the job done, and that is going to be our amino-acyl tRNA synthetases. We'll be talking about that more on the next slide, our ribosomes, and our mRNA strand. Like we mentioned before, that AUG codon is the start codon. So that is going to code for methionine and initiate translation. We also have a CCA amino acid binding end. You can see that right up over here. That is specifically going to bind the amino acids. And then we've already talked about the anticodon. Now I mentioned one of the important players here is the amino-acyl tRNA synthetases. And if you notice, that ends with aces, so we know that this has to be an enzyme. And this is going to help the tRNA get charged when it's in the cytoplasm. So if you remember in the previous section when we talked about pre-tRNA synthesis by RNA polymerase III, we were only creating the RNA portion. And so we need this tRNA to get charged, and it does this by getting linked to the correct amino acid utilizing this enzyme, amino-acyl tRNA synthetase. And the way the enzyme does this is by first attaching the tRNA. We can see the complementary anticodon here, and it binds it within its specific site here, and the amino acid that is going to match that anticodon. So in this case, it is cysteine. And so they enter the enzyme, and we are going to utilize ATP in order to do this. So we need to hydrolyze ATP and break off 2 phosphate bonds. We know that those are the high energy bonds, which then turns ATP into AMP and 2 inorganic phosphates. And now our amino acid is activated, and that AMP can leave, and the amino acid has been transferred to our tRNA and is ready to participate in translation. Now let's get started in talking about the actual process of translation. And we're going to start with initiation. We need to find our initiation complex. So we're going to look for that 7-methylguanosine cap at the 5' end, where the initiation complex can bind. We have our cap binding protein and other initiation factors as well that are going to help move our ribosome over to the 5' end. Our complex looks for that AUG start codon, and also we're going to find this sequence known as Kozak's rules found near that AUG in order to start. And we'll also have the R in here represent either an adenine or guanosine, but can't be C or U in that area. So then our ribosome is going to find the start codon and bind, and our MET tRNA, MET for methionine, remember that is going to be the amino acid for AUG, is going to enter an area known as the P site. This ensures that we are going to have this A site empty for another tRNA with an attached and activated amino acid to enter. Our next step is elongation, and we're going to see that elongation is pretty much the same within prokaryotes and eukaryotes. And we mentioned in the previous slide, we've got these 3 different sites within our ribosome. So let's go through them really quick. This first one on the right that we see is our A or amino-acyl site, and this is binding our incoming charged amino-acyl tRNAs, meaning that we've got that amino acid already attached and ready to go. The P site here is going to bind the charged tRNAs carrying the amino acid and form that peptide bond with our growing polypeptide. And then the E site is essentially for those tRNAs that don't have their amino acid on there anymore to exit. So they can drift off and be recharged with another free amino acid so it can come back when it is needed. And this little note is what I mentioned in the previous slide, how we are going to have that tRNA, that methionine tRNA enter the P site first. All right, let's continue elongation. We're going to see those charged tRNAs coming in, binding their amino acid to that polypeptide chain. Notice how this is shifting, and eventually that tRNA that entered the E site is going to leave. And this is known as the translocation event, how they move from the A site to the P site to the E site. And we induce these conformational changes in order to make that happen. We also have a ribozyme known as peptidyltransferase, and this helps to form that peptide bond between the amino acids in our large subunit. And in order to do this, we need some energy, right? So we are going to utilize GTP hydrolysis to make this happen, where we break off a phosphate bond and have it turn into GDP and that inorganic phosphate. Now for our last step here in termination. This is going to occur when we read that mRNA strand and we run into one of those stop or nonsense codons. And then we have no complementary tRNA to bring in additional amino acids, and our P site amino acid is going to detach from its tRNA and release the new polypeptide that has been created. So you can see that right over here where we do have that last tRNA binding its amino acid to the polypeptide, and we read that stop codon, and a releasing factor is going to be present to allow that tRNA to let go of the polypeptide. We will then see that our subunits of the ribosome will dissociate from the mRNA strand and from one another. The mRNA strand then is going to be degraded. We don't need it anymore, and we can release the nucleotides that were laying right along it so that we can create a new mRNA strand for further protein synthesis if needed. And the very last thing we're going to talk about is protein folding, modification, and targeting. We know that our amino acids can be chemically modified, there's signal sequences that are appended, and our new protein is going to fold into its 3D structure. You can kind of see the process taking place over here and it being released for modification. Now we do have something on here called a signal sequence, and that helps direct it to the specific cellular compartment that it needs to go to. You can kind of think of it as the protein's train ticket so that it can go to the proper destination. And when it arrives to that destination, then that sequence is going to be clipped off. And as this protein is traveling to that destination, sometimes it can start to aggregate. And so we can also have these helper molecules known as chaperones that can prevent that aggregation during the folding process. Now, if we do have any changes in temperature or pH, it could also cause it to misfold or even cause it to denature.